So basically, this is the transmitting neuron, which transmitted the impulse from the dendrite to the axon, and transmitted to the receiving neuron, which is called the postsynaptic cell neuron actually. And a neuron is made of cell body and axon. Dendrite is a part of the cell body and collected the impulse, and inside the cell body is the nucleus, and axon hella connected axon and cell body, and myelin sheath is covered the axon, and which is the other part is the knot of the revere, and impulse travels from the dendrite to the axon and to the receiving neuron.
So the stimulus is then picked up by the dendrite, which travels down the axon as an action potential and jumps across to the postsynaptic cell, which is the receiving neuron, and travels down to the next cell.
Right. Okay, so this is how the ion pumps and channels function with the action potential. So first, as the action potential goes through the axon, it activates a ion pump, which is shown here in purple, and it pumps a lot of sodium ions outside the cell and pumps a low concentration of potassium ions into the cell. And this thing causes a concentration gradient where there's a high concentration of sodium ions and a few potassium ions outside the cell, and a low concentration of mostly potassium ions inside the cell. And so this causes a voltage gradient where the outside of the cell is positively charged while the inside is negatively charged. And this causes the sodium ions to go through the sodium channel into the cell. And as it goes through, it activates more sodium channels along the way to bring more sodium ions into the cell. So, and then after that's done, after the sodium concentration is leveled, then the potassium ions inside the cell are diffuse through the potassium channels, and then they end up outside the cell. So again, it keeps doing that. It activates more potassium channels along the way as it diffuses outside the cell, and then it keeps doing it until the concentration levels off. And this process occurs through the axon, along the entire length of the axon, and it helps bring the signal through that length. So that's how the ion pumps help the action potential travel through the axon. This is what happens after the action potential is sent through. So after polarization, the concentration gradient has to be reset in order to prepare for another action potential. So here we have the sodium ion pump, which helps stabilize the concentration gradient and resets it. And then the sodium sodium ions and potassium ions are then mixed, so there's no concentration or voltage gradient at the moment. And then what happens is that the potassium ions are then pumped down, the sodium ions are pumped outside the cell, and whenever a new action potential comes through, it then activates each sodium and potassium channel along the way, and then restarts the process. So that's it. It's basically polarization and then the depolarization in order to prepare for the next action potential.
Okay, so this is the membrane potentials versus time graph. And this is the threshold, and the threshold is a level of stimulation required to trigger a neural impulse. And over here, this is the resting potential, and these are the two stimuli produced by hyperpolarization. Hyperpolarization is a change in the cell's membrane potential that makes it more negative. And this increases membrane permeability to the potassium ions. Also, large stimuli results in large hyperpolarization.
As mentioned previously, this is threshold, this is resting potential, and this is the depolarization, which means the reduction in the magnitude of membrane potential. Yeah. And depolarization produced by two stimuli that increases membrane permeability to Na+, and the larger stimuli results larger depolarization.
In this graph, the action potential is triggered by depolarization that reaches the threshold and it peaks.
Okay, so saltatory conduction is the mechanism in which the current produced during the rising phase of the action potential travels to the next node. This is where it depolarizes the membrane and regenerates the action potential.